Imagine trying to map like the exact location of a million different conversations happening in a crowded city.
Oh man, that sounds like an absolute nightmare.
Right. Now imagine you also have to record what every single person is doing for a living and somehow track whether the doors to their offices are locked or open.
Yeah, that's a lot of data.
And finally, imagine you have to do all of this simultaneously on a canvas the size of a pinhead.
Exactly.
Welcome to the deep dive. Whether you're actively exploring computational biology or you're just, looking to understand the invisible machinery of life, you are in the right place.
We are super glad you're here.
Today, our mission is to decode a framework called SMART, and that stands for Spatial Multi-omic Aggregation using graph neural networks and metric learning.
It's a mouthful, I know.
It really is. But by the end of this conversation, you are going to look at the very fabric of human tissue in a completely different way.
It really is just a mind-bending challenge. I mean, we are talking about the absolute frontier of biology right now, which is spatial multi-omics.
Which is relatively new.
Very new. Like for decades, if we wanted to know what a cluster of cells was doing, we basically had to throw the tissue into a blender and sequence the resulting smoothie.
A biological smoothie. Nice.
Right. So we got a fantastic list of ingredients out of it, but we completely destroyed the map. We had absolutely no idea where those cells actually lived in relation to one another.
So, yeah, you know the ingredients of the cake, but you have no idea how the layers were actually put together.
Precisely. But today, we have these cutting-edge platforms with names like 10x Genomics Visium or MyZarsec and StereoSciaSec.
Right, the big heavy hitters.
Exactly. And these tools allow us to measure multiple biological languages all at once, right, where the cells sit in their natural tissue architecture.
And what are those languages exactly?
Well, we're capturing transcriptomics, which is RNA. We're capturing epigenomics, which looks at chromatin accessibility, and proteomics, which tracks the actual proteins.
Okay, let's unpack this for a second because we just threw a lot of omics at the listener.
Fair enough, yeah.
Proteomics makes sense. I mean, that's mapping the actual proteins, the physical workers doing the heavy lifting in the cell.
Right.
Transcriptomics, the RNA, is kind of like intercepting the blueprints being sent to those workers.
That's a great way to think about it.
But what exactly do we mean by chromatin accessibility? Because that one always trips people up.
Yeah, so think of your DNA as an incredibly long, massive instruction manual.
Okay.
To fit inside a microscopic cell, that manual has to be spooled up really tight, wrapped around proteins to form what we call chromatin.
So it's packed away.
Exactly. And if a section of that DNA is spooled too tightly, the cell's machinery just can't physically read it.
Oh, I see.
So chromatin accessibility, which is usually measured by ATAC data, it tells us which pages of the instruction manual are actually open and available to be read by the cell.
That makes perfect sense. So taking millions of these different molecular data points, the proteins, the RNA blueprints, the open instruction pages, and somehow mapping them into a single unified picture without losing the details? I mean, how do you even begin to do that without completely crashing a computer?
Well, that is the core problem, because different omics have completely distinct mathematical shapes and statistical distributions.
They don't play nice together.
Not at all. Let's look at the RNA data, for instance. You might be tracking tens of thousands of genes across a tissue sample, but the data is incredibly sparse.
Meaning there's a lot of blanks.
Tons of blanks. You will see a massive amount of zeros. And here is the agonizing catch for biologists. Does a zero mean the gene is genuinely turned off in that cell?
Or did the machine just mess up?
Exactly. Did our chemical regions just fail to capture a faint signal? That's known as a technological dropout.
So it's like trying to listen to a radio station with terrible static. You don't really know if the DJ stopped talking or if you just lost the signal.
Yes. Perfect analogy.
And previous computational models basically failed at this, right? Like MISO or MOFA plus A. They either mashed the protein lists and the RNA lists together and just hoped for the best.
Or they totally ignored the spatial satellite map entirely. They just couldn't handle the where and the what's simultaneously.
Right.
And that is exactly where SMART steps in with its baseline mechanics. It starts with a two-step foundational process to basically filter out that static you mentioned.
Okay, what's step one?
Step one is principal component analysis, or PCA.
I've heard of PCA in data science, but how does it work here specifically?
Well, instead of feeding the neural network tens of thousands of raw, noisy data points filled with ambiguous zeros, SMART uses PCA to compress that high-dimensional data down into lower manageable dimensions.
So it shrinks it down.
Right. It looks for the main underlying themes or the principal components of the data. This prevents the computer from being fooled.
Because you don't want the model assuming two cells are biologically identical just because the sequencing machine failed to read their RNA and gave them both a bunch of zeros.
Exactly. You get a totally false positive. PCA distills the data to its most meaningful variations.
Okay, so step 1 shrinks and cleans the data. It cuts through the static. What is step 2?
Step 2 is where SMART brings the physical map back into play and uses KNN, or K nearest neighbors.
Okay, how does that work?
It builds a spatial neighbor graph using the actual X and Y physical coordinates of the tissue slice. It draws a mathematical line connecting a spot to its closest physical neighbors.
Literally drawing a map.
Exactly. So if the tissue is analyzed on a square grid, it connects to its 4 closest neighbors, so K equals 4. If it's a hexagonal grid, like the 10x Visium platform uses, K equals 6.
Okay, let's pause for our first mental checkpoint. For everyone listening, if you are tracking the logic here, think of it as 2 core bullets to remember.
Always good to recap.
Bullet 1. Spatial multi-omics is about capturing the RNA blueprints, the open DNA pages, and the worker proteins without destroying the neighborhood.
Spot on.
And bullet 2. SMART's first move is to use PCA to clean the static and KNN to literally draw the map connecting the physical neighbors.
Exactly.
So ask yourself this: if you were trying to map your own hometown and you connected all the houses by physical streets, how do you mathematically figure out the culture of each neighborhood? Because physical streets don't tell you what the people are actually doing inside.
Right, and that transition is exactly where the magic happens. Once SMART has this physical graph, it processes it using an encoder called GraphSage.
GraphSage.
Yeah, that stands for Graph Sample and Aggregate Convolution. When GraphSage looks at a specific spot in the tissue, it doesn't just evaluate that single spot in isolation.
What does it do?
It samples and aggregates the biological data from its physical neighbors, layering all that local context into a brand new unified profile called an embedding.
Wait, I'm getting lost here. If the algorithm is only looking at a cell's immediate physical neighbors to figure out what it is, doesn't that create a massive blind spot?
What do you mean?
Well, what if I have 2 immune cells that are doing the exact same job? They are functionally identical, but they are physically on opposite sides of the tissue sample. A purely spatial graph would completely miss their connection, wouldn't it?
You have just identified the fatal flaw of almost every earlier spatial model.
Really?
Yes. They become so utterly obsessed with physical proximity,
Yeah.
who is sitting next to whom, that they become completely blind to long-distance biological relationships.
So how does SMART fix that blind spot?
With a mechanism called metric learning and triplet loss.
Okay, I want to try an analogy here. Cosmetric learning and triplet loss kind of sounds like, I don't know, a bad indie band.
It really does.
Let's think of this like a social network algorithm. So imagine a specific spot in the tissue. We'll call this the anchor. That's you.
Okay, I'm the anchor.
Now, the algorithm uses something called mutual nearest neighbor matching to search the entire tissue for another spot that is basically your functional biological soulmate, even if they live in another city, across the tissue. That's the positive sample.
That is exactly right. That's the perfect match. And then the algorithm deliberately seeks out a negative sample.
The terrible match.
Right. It calculates the 60% of spots that are the furthest physical distance away from your anchor and randomly picks one. This is a spot that is highly likely to be completely biologically irrelevant to you.
So the algorithm has a trio, right? The anchor, which is you, the positive, your long-distance soulmate, and the negative, a completely irrelevant stranger. What does the math actually do with those 3?
The triplet loss function forces the graph neural network to adjust how it views the data. In the mathematical space, it actively pulls the anchor and the positive sample closer together.
Bringing the soulmates together.
Yes, while aggressively pushing the negative sample away by a specific rigid margin. In SMART, that margin parameter is called tau, and it's set to 0.5.
Oh, wow.
Yeah, this mathematically forces functionally identical cells to cluster together in the data map, no matter how far apart they are in the physical tissue.
Okay, I have to play devil's advocate here.
Go for it.
If we are aggressively squishing and pulling the data around just to make similar things touch in the computer's memory, aren't we basically hallucinating data? Like, how do we know we aren't completely warping the underlying biological reality?
It is a totally valid fear. And the developers of SMART actually built a failsafe specifically for it, called the reconstruction loss.
Okay, how does that work?
After the encoder creates these new unified profiles, a decoder runs in reverse. It tries to reconstruct the original raw PCA features for the new profiles.
Yeah.
It literally forces the model to prove its work.
Oh, that's smart.
Yeah, there's a tunable parameter called lambda that balances this. It literally weighs the spatial smoothing, the pulling and pushing of the triplet loss against the exact grounded reality of the reconstruction loss.
So it's constantly checking its own homework.
Exactly.
All right, time for our next checkpoint for you listening. Bullet 1. The GraphSage engine samples the local neighborhood so a cell understands its immediate surroundings. Bullet 2. The triplet loss mechanism acts like our social network algorithm, finding distant functional soulmates and pulling them together. And bullet 3. The reconstruction loss is the anchor to reality, ensuring the model doesn't just hallucinate patterns.
And to connect this to why you should actually care, imagine you are analyzing a scattered cancer tumor.
Okay.
Tumors are not neat, polite little spheres. They are chaotic, They invade, they scatter, and they break apart.
Right, they're messy.
So if your computational model cannot link distant rogue cells that are acting the exact same way, you fundamentally misunderstand the pathology of the cancer.
Which brings us from the theory into the actual biology, because it is one thing to have elegant math, it is another entirely to see it work on real tissue.
Exactly. And the researchers first proved the concept on simulated triomics data, where SMART preserved the original distances better than anything else. But the real test came with case 1, the human lymph node.
Right, using that 10x Visium data we mentioned earlier.
Yes, mapping both RNA and proteins. And the lymph node is a structural nightmare. You have fatty tissue on the outside, you've got the cortex layer, and deep inside, these incredibly delicate web-like structures called medullary sinuses and cords.
And SMART successfully mapped 7 distinct anatomical regions here. It didn't just, guess where things were. In the cortex, it correctly found high levels of T-cell markers, specifically the CD3E protein and the CCR7 gene.
Right.
But the real reason this is impressive isn't just that it found the T-cells. It's what happened in the deep center of the node, the medulla.
Right. Competitor models like Mephisto and CellCharter completely failed here.
What happened?
They oversmoothed the data. They basically took the delicate medullary sinuses and the medullary cords and just blurred them together into one giant blob.
Because they prioritized physical closeness over biological truth. They just assumed that if cells were next to each other, they must be the same.
Exactly. But SMART utilized the expression of a specific gene marker called CCN1. Because of its triplet loss and reconstruction loss balancing act, it held on to the specific reality of that CCN1 gene, and it perfectly drew the razor-thin boundary of the medullary sinus.
That is incredible. Let's escalate this, though. A lymph node is relatively static. What happens when the tissue is a moving target, like an embryo actively growing and changing shape?
That is a great question.
Which brings us to case 2, the developing mouse brain.
For this, they used MISAR-seq data on embryonic mouse brains from day 11 to day 18.5 of development.
Okay.
Here, SMART fused the RNA blueprints with ATAC data, that chromin accessibility we talked about earlier. And as the brain develops, structures are moving, folding, and differentiating at light speed.
And SMART was able to accurately map massive regions like the dorsal pallium. But what really caught my eye was this tiny, delicate structure called cartilago 1. Almost every other computational model completely missed it.
Because the genetic whisper of that tiny piece of cartilage forming was just drowned out by the noise of the massive brain tissue surrounding it.
Right.
But SMART's dual engine caught the whisper. It detected the subtle epigenetic shifts, the opening of specific pages in the DNA manual that actually defined that cartilage.
Amazing. Let's do another quick mental recap. Bullet 1. SMART doesn't just work in theory, it outperforms other models on real, complex dual omics data, keeping anatomical borders super sharp.
Yep.
Bullet 2. It successfully mapped complex immune zones in lymph nodes and detected hidden structural whispers in developing brains. So, for you listening, think about this. If you are a pathologist staring at a tissue slide, SMART acts like a set of biological superglasses.
I like that. Super glasses.
Right. It doesn't just show you the cells, it highlights the invisible molecular borders driving disease or development. So how much invisible structure exists right beneath the tools you use every day?
The amount of invisible structure is just staggering. And the more we realize is there, the more data points we try to measure, which introduces the next massive hurdle in spatial biology scale.
Right. What happens when our map gets overwhelmingly massive? We are talking about scaling up to epic speeds, moving from dial-up internet in biology to fiber optic.
To test this, the researchers ran an extreme stress test. They took a slice of a mouse spleen and analyzed it using stereo CIT-ESEC.
Which is incredibly detailed.
Yeah, this technology provides subcellular resolution. They processed the data at what they call bin 10 resolution, meaning they were looking at grids that were just 5 micrometers wide.
Oh, wow.
To put that in perspective, at that resolution, they generated 756,430 individual spots of data on a single tiny slice of tissue.
Over 750,000 spots. So what happened to the competitive models when they tried to process that?
It was a computational bloodbath. Every other model either crashed completely or simply ran out of GPU memory by the time they hit 190,000 spots.
They couldn't handle the scale.
No. They tried to compute the entire global map of the tissue all at once, and their memory just folded.
But SMART processed all 750,000 plus spots, and it did it in 56 seconds.
56 seconds. It's wild. And that is the brilliance of the graph SAGE architecture. Because it only samples local neighborhoods instead of calculating the whole universe simultaneously, its memory footprint is incredibly light.
Okay, 56 seconds for 2D tissue is unbelievable. But life isn't flat, right? If we really want to understand a tumor or an organ, we need 3D models. We need multiple slices stacked on top of each other.
And that requires a variant of the framework called SMART-MS, which stands for multi-section. But when you start stacking multiple slices of tissue, you run into a nightmare called batch effects.
Okay, let me guess. If I take 2 consecutive, almost identical slices from the exact same human tonsil, but I sequence slice A on a Tuesday and slice B on a Wednesday, the machine might read them differently.
Exactly. Simply because the temperature in the room was slightly different or the chemical region sat out for an hour longer, the sequencing machine will introduce artificial differences.
That's so frustrating.
It is. The computer will look at 2 identical cells in slice A and slice B and think they're totally different types of cells just because of technical noise.
So how on earth does SMART-MS fix artificial batch effects?
First, it utilizes an algorithm called Harmony. Harmony is specifically engineered to perform low-dimensional embedding and batch alignment.
Meaning it smooths out the dates.
Right. It mathematically calculates those artificial batch differences, the Wednesday versus Tuesday noise, and iteratively adjusts the data. It preserves the true biological variation while erasing the technical static.
Okay.
And once Harmony cleans the data, SMART-MS does something absolutely brilliant to build a 3D map. It uses cross-section triplets.
Wait, is this our social network algorithm again, but now it's matching people across different moments in time or different layers?
Precisely. When constructing the triplet loss we talked about earlier, the anchor spot is in slice 1. But the algorithm deliberately searches for the positive, match the functionally identical biological soulmate in slice 2 or slice 3.
Oh, that is so clever.
Yeah, and the negative sample is still drawn from the anchor's own slice to maintain contrast.
By forcing the model to match anchors to positive samples in completely different slices, it is literally using the biology to stitch the 3D physical map together.
Exactly. And we saw this flawlessly executed in case 3, the human tonsil. Tonsils are incredibly dense, complex organs packed with lymphocyte clusters.
Right.
SMART-MS analyzed 3 separate slices of a human tonsil and perfectly aligned them. It tracked the germinal centers through all 3 slices using the CR2 protein marker. It tracked the follicles using the CD19 marker and the T cell zones using CD3E and CD4.
And it did all of this while vastly outperforming other 3D batch correction models. Models like Total 6 and Present BC either failed to accurately detect the structural zones or they completely blurred the boundaries between the B cells and the T cells.
Yeah, SMART-MS maintained the absolute highest biological preservation scores across the board.
It decisively proves that we can integrate massive, multi-slice, multi-omic data sets without sacrificing the granular, microscopic reality of the tissue. Time for our final recap. Bullet 1. SMART is computationally elite. It processes 750,000 data spots in under a minute, while competitor models literally crash and burn.
They really do.
Bullet 2. To build 3D maps, the SMART-MS variant uses the Harmony algorithm to erase technical batch effects and then uses cross-slice triplet matching to stitch multiple layers into one cohesive biological reality.
And this raises a really important question for you listening, regardless of your specific field. What massive computational bottlenecks in your own work could be solved with a more elegant, localized sampling approach.
That's a great point.
We so often try to force our computers to ingest the whole universe at once, when sampling the local neighborhood like GraphSage does is infinitely more efficient.
So what does this all mean? We have covered a massive amount of ground today, from shrinking data with PCA to finding biological soulmates with triplet loss to building 3D models of human tonsils.
Ultimately, the profound value of SMART is that it isn't just a rigid algorithm. It's a completely flexible, modality-independent framework.
Right, it adapts.
Exactly. Whether you are using spot-based data like 10x Visium or single-cell segmented data from high-resolution imaging, SMART adapts. It takes wildly different biological languages, the RNA blueprints, the protein workers, the open chromatin pages, and translates them into a single unified map.
And it achieves this by perfectly balancing local geography with long-distance functional relationships.
Exactly.
It's like we finally have a universal translator for the tissue microenvironment. It doesn't just show us a list of what is there, it shows us exactly how it is organized, seamlessly mapping the delicate borders of disease and development.
And if you extrapolate that out, it leads to a truly fascinating implication. We've seen SMART master spatial relationships in 2D, and we've seen it stack slices to master 3D across multiple omics.
Yeah.
But if this framework can align completely different molecular languages across spatial coordinates so flawlessly, what happens when we introduce a fourth dimension?
You mean time?
Exactly. Imagine applying this exact cross-section triplet loss matching, not just across static slices of a tonsil, but to track how a cancer tumor microenvironment mutates across time during a prolonged course of chemotherapy.
Oh, wow.
How would the ability to track those temporal spatial changes at a subcellular resolution revolutionize the future of personalized medicine?
That is exactly the kind of question that pushes science into the future. For you listening, keep questioning the invisible patterns around you. Look a little closer at the borders and boundaries you take for granted. And thank you for taking this deep dive with us.